Hey, you guys. Sorry I can't be there today. I have to have a root canal, so consider yourself lucky. You get to learn about genetics, and I have to have a root canal. Anyway, I wanted to give you a little overview about this guy over here, Gregor Mendel. He's known as the father of genetics. He was around from 1823 to 1884. Um, and as I think I mentioned before, he was an Augustinian monk and he wandered around the gardens of his monastery and he wondered about plants that looked different or atypical from the other plants. And he ended up writing a book called Experiments with Hybrid Plants and came to a bunch of conclusions. And you might be wondering, why did he choose peas? Well, first and foremost, they're pretty easy to grow. And they're fast growing. I don't know if any of you ever did little pea experiments as a kid, but they grow fast. And he could grow a lot of generations in the 8 years that he was carrying out this study. They also self-fertilize, so you don't need a lot of plants. You can have them cross with each other, and they don't need a mate. But some of the traits he was looking at, you can see down here, texture of the seeds, color of the seeds. He didn't want to study traits that had continuous variation. So if you think about the traits we did in class the other day with tongue rolling and hitchhiker's thumb, they were either or study characteristics. They didn't have continuous variation, and we'll talk more about that later. So he wanted traits that had either or form, so whether the plant was short or tall, smooth or wrinkled, green or yellow pods and seeds, all those things. And here's a little overview of his experiments. First, he wanted to create sort of purebreds or true breeding varieties. Remember when we talked about homozygous? So he wanted them to be homozygous dominant. So for every trait, and this took years because he didn't want any random recessive ones. And then he would find an atypical variety, so the homozygous recessive, and transplant them next to the typical variety and kind of just look at what progeny or offspring arose. And he usually found that the offspring all looked like the parent with the dominant trait. So he found that all the offspring retained certain essential traits from the parents. So here's a cross that he did where he crossed wrinkled plants right there and smooth plants right there. Sorry, my battery was just dying. So anyway, he wanted to cross pure smooth plants with random wrinkled ones that he saw. So he wondered if the offspring of these two plants would be smoothish rough, or how they would look. He wondered if some of them would be smooth and some would be rough. But he found that the next generation of plants produced only smooth plants that looked like the original sort of dominant one. But then he crossed those smooth ones, he self-fertilized them, so he had them cross with each other. And the next generation, he got a different variety. So when he crossed these two together with that genotype, big S, little s, he got all smooth ones. But then when he crossed those together, he got mostly smooth, but that rough one showed up again. So here's an example of color cross. So he took a bunch of yellow peas and he crossed them with green peas, which were the recessive one. And you can see here that all of them became yellow. So we talked a little bit last class about Punnett squares, but they're a great way to predict the offspring that certain parent crosses will give you. And it's a cool visual form to do it. So here's a purple heterozygous flower crossed with a purple heterozygous flower, and you can sort of fill in the chart. So here there's two little peas, and this would be big P, big P, big P, little P, big P, little p as well. It's sort of like you could imagine doing a foil, first, outer, inner, last, which is a little bit more complicated, but it's just, I think, a cool way to see offspring. So there again, you've got a heterozygous female or male crossed with a heterozygous purple pea plant, and that's what you get in the Punnett square. Tongue rolling, you can make Punnett squares for these. So Dolly can't roll her tongue, so we know she's homozygous recessive for that trait. Phenotypically, she's a non-roller. We know her genotype is little t. Dolly's dad can't roll his tongue either, so we know he's phenotypically a non-tongue roller, and his genotype is little t. But Dolly's mom can roll her tongue, so her phenotype is that she's a roller, but we don't know if her genotype is homozygous dominant or heterozygous. So in order to have a daughter who cannot roll her tongue, her mom has to be heterozygous. And if you look here at this Punnett square, in order to have a daughter who can't roll her tongue, she had to get a little t right there. Some alleles are related through incomplete dominance. So here's a red snapdragon crossed with a yellow snapdragon, and you get all pink snapdragons. So it's kind of a blending of the two. But again, in Mendel's case, we used complete dominance. So that's a pattern of gene expression where the phenotype of the heterozygous individual is intermediate between the others. So when you're doing genetic problems, I want to tell you about a couple of generalizations. First, you always have to have two alleles for a trait. So the genes are usually symbolized by the first letter of the dominant gene. That said, sometimes capitals look the same as lowercase, so sometimes we change that. But the letter for the dominant gene is always capitalized, and the letter for the recessive trait is always lowercase. So just make sure you can tell the difference between the two. T's are a great one. C's and W's are not. Wild type is the typical form of the organism, strain, or gene, whereas pure is a trait for those with identical genes, so homozygous. Hybrids are what we call mixed genes or heterozygotes, and gametes only carry one allele for each trait, so because they're haploid. So after all of Gregor Mendel's trials with pea plants over the course of 8 years, he came up with some rules that I just want to tell you about. There's 3 of them. His first rule is the rule of dominance and recessiveness, and with this he said that the allele that's expressed in the phenotype when the genotype is heterozygous or hybrid is the dominant allele. So the allele that's masked is the recessive allele. For example, tongue rolling. So here's just a little explanation of that. So he says here, I crossed purebred green and purebred yellow plants. Surprisingly, all of the offspring were yellow. The green phenotype has disappeared. I allowed the yellow plants to self-fertilize to see what the second generation would look like. Some of the second generation offspring were green. After performing many experiments, I figured out the basic rules of inheritance and why the green color reappeared. Remember, I found the pea color is controlled by one gene, which has a green form and a yellow form. Each form is called an allele. I'm homozygous. I have 2 green alleles. I am homozygous. I have 2 yellow alleles. When a purebred green plant is crossed with a purebred yellow plant, the offspring inherit one allele of the color gene from each parent. Plants with 2 different alleles of a gene are heterozygous, and since all the heterozygous offspring are yellow, then the yellow must be the dominant over the recessive green allele. The green allele didn't disappear. Its effect is not seen in a heterozygote. Peas are yellow when they have either 2 yellow alleles or 1 yellow allele and 1 green allele. Green peas must have 2 copies of the recessive green allele. Now, let's see how to sort out when we cross how they sort when we cross 2 heterozygotes. When 2 heterozygous plants are crossed, the resulting progeny mostly show the dominant phenotype, but the recessive phenotype reappears. Ta-da! So the next rule he has is the rule of incomplete dominance. So when the gene is a heterozygous zygote in incomplete dominance, it results in a phenotype which is a mixture of the 2 genotypes. So an example of this would be the red and the pink snapdragons. Rule of segregation is that during meiosis, two alleles of a gene separate during the formation of the egg and the sperm, and they separate segregate independently of each other. So here's another one showing that. And this is Reginald Punnett. William Bateson and I were very keen on Mendel's laws of inheritance. Herr Mendel was correct about how parental genes are passed on to offspring. He was also correct about the 3-to-1 ratio of dominant to recessive traits in a hybrid cross. Let's show you what happens in these crosses. Each trait is defined by a pair of genes. Each parent can produce 2 types of gametes. One allele is randomly separated into each gamete. This is Mendel's law of segregation. I invented the Punnett square to keep track of the alleles in the gametes. So in Herr Mendel's heterozygous cross, the allele of one parent are represented along the top and the other along the side of the square. See how they pair up in the empty boxes representing the offspring? Offspring result from pairing of the parental gametes, one for each parent. The offspring genotypes are seen there. Of course, little y peas are green. And while big Y and big Y, little y peas are different genotypes, they also have yellow phenotypes. As you can see, there are 3 yellow pea plants to 1 green pea. This is Mendel's 3-to-1 ratio. My squares are especially useful when tracking the inheritance of more than one trait, but we'll do that later. So the law of independent assortment is that alleles of one gene separate independently of the alleles of any other gene. In other words, the way that one pair of genes segregate has nothing to do with the way the other pairs segregate. And that's it for Mendel. And here's, just to end things up, here's, oh, that link didn't work. Anyway, I hope you liked this show, and good luck on your notes and working on questions 1 through 12.